Hey everyone, and welcome back to The Explainer. So, if you are a focused learner, you're going to want to buckle up because today we are diving into an absolute masterclass. We're tackling one of the most honestly overwhelming topics in biology, the animal kingdom. Look, if you've ever felt totally lost trying to memorize just endless lists of biological features, you're in exactly the right place. We are going to break this whole thing down using clear, incredibly structured blueprints. Just to wrap our heads around the sheer scale of the problem we're solving today, take a look at this number. Scientists have actually identified somewhere around 1.7 to 1.8 million living species on Earth. And out of those, more than 1.2 million are animals. Yeah, over 1.2 million distinct animal species, literally scattered all over the planet. So, the million-dollar question here is, how on earth do we possibly sort 1.2 million different species without completely losing our minds? Think about it. If we just dumped a million animals into a giant room, studying them would be impossible, right? We have to classify them. We absolutely need clever frameworks and foundational biological rules to organize all this chaos into neat, easy-to-learn categories. Here is our game plan for today. First, the animal sorting problem. Next, the body blueprint. Then some classification cheat codes. And finally, a case study on phylum Porifera. All right, let's jump right into section 1, the animal sorting problem. To actually sort animals, we really have to look at how life organizes itself right from the absolute ground up. It's kind of like building a city, you know? You start out with loose cells just sort of working independently. That's the cellular level, which we see in the simplest animals. Then those cells start teaming up into specialized groups, giving us the tissue level. Upgrade a bit further, and those tissues form distinct organs, like you see in flatworms. And finally, organs start collaborating in these big, complex networks, the organ system level. And that right there is what we humans and pretty much most higher animals operate on. Moving right along to section 2, the body blueprint. Okay, so aside from cellular organization, scientists look at core physical features, basically an animal's internal plumbing, to categorize them. First up is digestion. Really primitive animals use what's called a blind sac body plan. This is an incomplete digestive system, where one single opening literally serves as both the mouth to take in food and the anus to expel waste. I mean, talk about multitasking, right? On the flip side, more advanced animals have a complete digestive system. It's essentially a tube-in-tube plan with two completely separate openings. And this major upgrade kicks in starting with the roundworms or Ashkelminthus. Now let's talk about the rest of the internal plumbing, specifically the circulatory system. In open circulation, blood just flows freely in open spaces. It's literally bathing the tissues and cells directly. There are absolutely no tiny capillaries. But in a closed circulation system, which is what earthworms and, we humans have, the blood is strictly confined to a highway of vessels, arteries, veins, and capillaries. It's just a highly, highly efficient and organized setup. And this complexity actually builds right from the embryonic stage. An animal's body is largely defined by its germ layers. Diploblastic animals are on the simpler side. They develop from just two layers, an outer ectoderm and an inner endoderm. Triploblastic animals, which covers everything from flatworms all the way up to humans, add this absolutely crucial third layer right in the middle, the mesoderm. This middle layer is basically the secret ingredient for building highly complex organs. Now, understanding the colon is an absolute milestone in biology. Seriously, pay close attention to this one. The colon is a fluid-filled body cavity located right between your outer body wall and your inner digestive tube. But here is the critical rule. To be a true colon, this cavity has to be completely lined by that mesoderm on both sides. It's this beautifully secure space where your major internal organs can just safely reside. So, based on that exact rule, animals fall into 3 cavity buckets. Acoelomates have 0 body cavity. They are just packed solid. Pseudocoelomates have a false cavity that appears in these scattered pouches because it isn't perfectly lined by mesoderm, and this weird middle ground is unique to the Ashkelminthes. Finally, we've got the colomates. These are the animals with the true, fully lined cavity, a feature you'll find in annelids and literally all higher life forms. All right, on to section 3, classification cheat codes. Okay, this is honestly my favorite part. We're shifting away from the dense biological theory and jumping into some exclusive, highly effective memory tricks to make studying this entire kingdom practically effortless. First cheat code, digestion. Just remember the simple formula IB3. The I stands for intracellular digestion, which applies strictly to the very first phylum Porifera. Food digests completely inside the cells. B3 stands for both types of digestion, and that applies to the next three phyla, Coelenterata, Ctenophora, and Platyhelminths. And as for the rest, literally everything else in the animal kingdom utilizes strictly extracellular digestion, digesting food inside a dedicated system exactly like we do. Next up is fertilization. Just memorize these two quick catchphrases, CC Hemazico and BAM. So CC Hemazico covers Coelenterata, Ctenophora, Hemichordata, and Echinodermata. All these guys rely entirely on external fertilization, meaning they just release their gametes straight into the water. BAM stands for both internal and external, which you'll find in Annelida and Mollusca. And the rest, every single other non-chordate utilizes internal fertilization. Boom, no brute memorization needed. For development, we're going to use the A3 rule. Whenever you're asked if an animal develops directly into a mini-adult or if it goes through a distinct larval stage, just look at the A3s. Aschelminthes, Annelida, and Arthropoda. These three specific groups can actually exhibit both direct and indirect development. All the rest of the non-chordates, they strictly use indirect development, meaning they are always going to pass through a distinct larval stage. Easy, right? Okay, let's bring it all together in section 4, case study, phylum Porifera. We are going to apply all these blueprint rules and cheat codes to the absolute simplest animals on Earth. Meet the sponges. Now, the name Porifera literally translates to pore bearers. And yeah, they are covered in tiny holes, they live mostly in salty marine environments, and despite the fact that they've been surviving for hundreds of millions of years, they still operate at the most primitive cellular level with these totally asymmetrical bodies. But hey, do not let their simplicity fool you for a second. Their water canal system is basically like a bustling city infrastructure. Check this out. Step 1, water enters through thousands of these tiny inlet doors called ostia. Step 2, all this water pools into a giant central cavity called the spongiocele. And step 3, the water gets pushed out of a massive exit door right at the top, which is known as the osculum. So you might be wondering, what's actually driving all this water flow? Enter the choanocytes, or collar cells. This is essentially the biological engine room. These highly specialized cells line that entire central cavity, and they have these tiny whip-like tails called flagella. By constantly, and I mean constantly whipping these flagella back and forth, they generate the continuous water current that literally keeps the entire sponge alive. And this constant flow of water just perfectly illustrates why that current is a matter of life or death. Because sponges completely lack complex organ systems, this flowing water literally does everything for them. It brings in tiny food particles for that intracellular digestion we talked about. It brings in fresh oxygen for respiratory gas exchange. It acts as a built-in sewage system, just flushing out excretory waste. It even transports gametes for reproduction. So their water canal system is their digestive, respiratory, and circulatory system all rolled into one beautiful package. Sponges also pack some pretty fascinating anomalies. For skeletal defense, instead of having bones like us, they're structurally supported by either these incredibly tough spongin fibers or these super sharp, needle-like spicules made of calcium or silica. And when it comes to reproduction, well, apart from regular sexual methods, they can actually reproduce asexually. They do this by fragmentation or by producing these highly specialized, incredibly tough internal survival buds called gemmules. To round off our case study, let's look at the 3 must-know sponge examples. First up is Cycon, which is also known as Sypha. Next, we have Eusmongia, famously known as the true bath sponge. Yeah, the kind historically used for literally washing up. And finally, Spongilla. Now, pay special attention to Spongilla. It is a massive, unique exception, because unlike the vast majority of sponges that live out in the salty ocean, Spongilla actually thrives in freshwater. So we've successfully decoded the biological blueprint of the absolute simplest animal, the sponge. And we've learned the structural frameworks that govern all 1.2 million species on Earth. Now, I want you to look down at your own hands. Just think about your complete digestive tract, your perfectly closed circulatory system, your triple-blastic germ layers, and that true column sitting inside you. Given all these incredible, highly advanced biological cheat codes we just explored together, where exactly do you fit into this massive evolutionary puzzle? Keep that thought in mind, keep that curiosity going, and I will see you in the next explainer.